Familial hypercholesterolemia is a genetic disorder that causes dangerously high levels of LDL cholesterol in the blood, significantly increasing the risk of cardiovascular disease for those affected. This condition follows an autosomal dominant pattern of inheritance, meaning if a parent has it, there is a 50% chance they'll pass it on to their child. The cause of familial hypercholesterolemia lies in mutations in key proteins like LDLR and the ApoB protein, which are involved in the process of cholesterol metabolism. Mutations in these proteins prevent the body from efficiently removing cholesterol from the bloodstream, leading to abnormal cholesterol buildup in the skin, tendons, and coronary arteries. Over time, this increases the risk of early heart disease developing even at a young age. But the LDLR and ApoB mutations are not the only two proteins affected. Variants in the PCSK9 gene can also contribute to the disorder. Normally, PCSK9 helps regulate cholesterol levels, but when mutated, it disrupts this process, further raising cholesterol levels in the blood. It is important to note that familial hypercholesterolemia is primarily caused by genetic factors. Environment or lifestyle changes cannot alter the inherited gene mutation, hence the term familial. However, doctors recommend lifestyle adjustments such as a low-fat, low-sugar, and low-alcohol diet to help manage the condition and reduce its impact on heart health. Understanding familial hypercholesterolemia is key to early detection and prevention because if we can interfere early enough, it can reduce the risk of heart disease and improve longevity and quality of life.
23andMe is a biotechnology company that conducts genetic tests that search for single nucleotide polymorphisms, otherwise known as SNPs, insertions, and deletions. These variations can impact the expression of genes. One gene that 23andMe tests for is the gene that codes for LDLR receptor proteins. Variations in this gene impacts the production of proteins that regulate and remove cholesterol from blood through its ability to bind to excess LDL cholesterol particles and take them to the liver. This gene is on the 19th chromosome and is typically expressed in the epithelial cells of the liver as well as on the outside of other tissues. Certain variants of this gene reduce the number of LDL receptors made or completely disrupt LDL receptor function. This leads to excess LDL in the blood, which is known to be a leading cause in higher chances of cardiovascular disease and heart attacks.
Our test will include hundreds of genetic variations in the PCSK9 gene on our S&P chip test. A specific genetic variation that we will include on our test is the D374Y mutation. This variation is a good indicator of genetic risk for developing this trait. The D374Y variation occurs at codon 374, where aspartic acid is replaced with tyrosine. This mutation enhances the binding between PCSK9 and LDL receptors. This increased binding leads to less LDL receptors being available in the liver. LDL receptors, which are responsible for cleaning cholesterol from the bloodstream, are less able to perform their normal function, leading to higher cholesterol levels. The D374Y mutation is linked with familial hypercholesterolemia, which is characterized by high cholesterol levels and a greater risk of developing heart disease, especially earlier on in life. The PCSK9 protein is made in liver cells. It makes sense that a mutation in the PCSK9 protein would lead to an increased cholesterol level since it is found in the liver, whose primary function is to process food.
While there are significant benefits to taking our test, there are some things to consider first. Discovering your genetic predisposition for familial hypercholesterolemia can only confirm a high cholesterol diagnosis, but not all people with familial hypercholesterolemia have high cholesterol. False positives and false negatives are very possible with these kinds of tests. Our tests aim to identify genetic variations to the PCSK9 gene, such as the presence of the D374Y mutation, which only produces a possibility that the PCSK9 gene's binding interaction will be increased from 10 to 25 times. This can potentially be significant, but does not guarantee a diagnosis. The process of being tested for a genetic disease, especially familial hypercholesterolemia, is both financially and emotionally straining. There is significant stigma surrounding most genetic diseases, but especially high cholesterol, as many view the disease in a very negative connotation. Additionally, worries about health complications can cause emotional stress, such as hypercholesterolemia can lead to dangerous heart conditions. Knowing you are positive may also cause issues to arise relating to food and eating disorders, as well as strain familial relationships. Data privacy should also be considered, and though we do our best to protect your data, it is impossible to guarantee